Hello, my name is Paloma Buceta.
And I'm Tiara Adler.
And we are from Seattle, Washington.
The rising threat of malaria. BioExpo's SeaVuria category involves a partnership between students in Seattle and Vuria, Kenya. Malaria is caused by the parasite Plasmodium, carried by female Anopheles mosquitoes. Malaria can be spread through a bite from an infected Anopheles mosquito, as well as contaminated blood and syringes, pregnancy before or during delivery, blood transfusions, and organ transplants. There are over 100 different Plasmodium parasite species. The four most frequent are Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, and Plasmodium ovale. Plasmodium falciparum develops quickly. It causes the most deaths in Africa, but it is typically curable. Plasmodium vivax is more widespread than other species. However, it produces less life-threatening symptoms and relapses can occur. Plasmodium malariae causes common malaria symptoms, and it can stay in the bloodstream for decades without even showing a symptom. Plasmodium ovale is quite rare, as it is seen mostly in West Africa and can cause relapses. The Plasmodium parasite reproduces sexually in mosquitoes and asexually in humans by first invading liver cells, dividing 40,000 times, and then invading red blood cells. The Anopheles mosquito bites an individual, taking in blood while injecting saliva with the parasite. When first in the bloodstream, the parasite is in its sporozoite stage. The sporozoite occupy the liver cells and develop into a schizont within a few weeks. The schizont contains thousands of small round merozoites. Once the schizont mature, the merozoites explode into the bloodstream, taking over the red blood cells. On the other hand, Plasmodium vivax and Plasmodium ovale have different stages. Once the sporozoite are in the bloodstream, they form into hypnozoites, which can become dormant for months or years. If activated, the hypnozoites develop into schizonts, and they can then cause relapses. Once the parasite first enters the bloodstream, both humoral and cell-mediated responses are triggered. The first immune response is when the sporozoites are injected into the bloodstream and antibodies are released to prevent them from traveling to the liver. If the parasite travels to the liver, cytotoxic T lymphocytes are sent to kill infected hepatocytes. Following that, antibodies work to block infection in the red blood cells from the merozoites. The final immune response is the carrying out of the infected red blood cells to the spleen, where they can be filtered out.
Following the infected bite of the Anopheles mosquito, the disease enters an incubation period, lasting anywhere from 7 to 30 days. This period of time between infection and the appearance of initial symptoms is often the leading cause of misdiagnosis. There are commonly two categories of diagnosis: uncomplicated and severe. Uncomplicated malaria is rarely observed and only lasts between 6 and 10 hours. Symptoms of uncomplicated malaria include a cold stage, a hot stage, and a sweating stage. The combination of fever, sweats, chills, headaches, nausea, and vomiting can seem like influenza symptoms. Diagnosis of uncomplicated malaria is dependent on evidence of parasites through microscopy. Conversely, severe malaria occurs when symptoms are complicated by organ failure and abnormalities in blood or metabolism. Additionally, malaria has the potential to relapse. In Plasmodium vivax and Plasmodium ovale infections, the dormant liver stage parasites, hypnozoites, may reactivate, causing symptoms any time within months, even years, after first exposure. The disease's manifestation depends predominantly on climatic factors such as temperature, rainfall, and humidity. Transmission will not occur in deserts or at high altitudes. Generally, malaria is transmitted at high concentrations in warm regions near the equator. The parasite Plasmodium cannot complete its growth cycle inside the mosquito unless temperatures are above 20 degrees Celsius or 68 degrees Fahrenheit. The parasite's short reproductive cycle makes malaria highly transmittable. The disease's success depends on a population's ability to combat or treat malaria.
Although there is no available cure or vaccine yet, there are many current treatments and preventions available. Treatment of malaria is dependent on a patient's conditions, including the type of malaria parasite the patient has, the severity, their age, and whether or not they are pregnant. There are many anti-malarial drugs on the market today. These drugs target the parasite in the blood. Other modes of prevention include spraying insecticide on walls, bed nets with or without insecticide, covering skin, and spraying skin and clothing.
The connections between malaria and poverty are definite, but high concentrations of malaria in poor countries are not a specific consequence of poverty. The location and severity of malaria are mostly products of climate and ecology, but those populations more affected by the disease tend to face the economic burden as well. The disease relies on various factors for maximum efficiency, all of which the village of Vuria supplies. In sub-Saharan Africa, agriculture accounts for 70% of the economy. Irrigation is a key component of the agricultural system, and water is conducive to the reproduction of mosquitoes. Rice is a staple crop in sub-Saharan Africa, but the crop relies on flooded paddies perfect for the breeding of malaria-potent mosquitoes. Additionally, mosquitoes feed on fruit nectar such as maize pollen. Treatment can be costly and leave lasting impacts. The girls from Bura have all experienced the effects of malaria and have seen what it does to the community. Deaths from malaria are common, especially in children. This preventable death can be very tolling for the families. Malaria can have impacts in education. Its symptoms prevent individuals from attending school, thus impeding on their chance to succeed academically. There also is a large unawareness of the disease within the Vuria community that makes prevention very difficult. Things like not taking anti-malarial drugs and the misuse of bed nets are just some examples.
Over the years, funding for malaria has increased. In order to have the best international resources and prevention, we will need an estimated $5.1 billion annually between 2011 and 2020. Unfortunately, that goal has not yet been met. The United Nations International Children's Emergency Fund has been a large contributor to malaria funding over the years. UNICEF has donated $1.57 billion to malaria control and child survival programs. Locally, the Bill and Melinda Gates Foundation has greatly helped fund and contribute to the malaria cause. With almost $2 billion grants to fight malaria, they provide 50% of funding for malaria worldwide.
Seattle Biomed is one of the largest malaria research centers in the U.S. Their focus is on discovering a vaccine against malaria for pregnant women, children, and people with liver stage malaria. In recent research, they have discovered the genomic sequence of Plasmodium falciparum. Researchers at the University of Copenhagen have discovered the endothelial protein C receptor, or EPCR, which aids the adhesion of red blood cells in the brain, which cause cerebral malaria. This discovery could be the gateway to prevention of this adhesion, allowing for more efficient blood flow. Identifying additional receptors that work congruently with EPCR could further yield advancement in unraveling the complexity of malaria. In 2010, they developed a vaccine from a weakened form of the malaria parasite called genetically attenuated parasite, or GAP, by deleting 2 important genes. The GAP vaccine was tested on mice and was proven 100% protective and accurate. The vaccine was then moved to human trials, only to be partially effective. They believe that the late liver stage GAP vaccine will be more effective because it allows the immune system more time to recognize the parasite's antigens before entering the bloodstream. We interviewed Dr. Alexis Kashansky, a senior scientist at Seattle Biomed. She focuses on the prevention of liver stage malaria. When asked about the GAP vaccine, Kashansky told us that the most recent trial, conducted in January 2015, tested the new version of GAP vaccine on 10 volunteers. The results have not yet been disclosed, but the new vaccine includes a 3-gene deletion out of 5,500 genes in the parasite. Due to the recent discovery of the EPCR, Seattle Biomed hopes to discover how to block the EPCR and other related protein receptors from adhering the infected red blood cells to the brain cells. We had the opportunity to meet our collaborating partners, Gloria, Viviane, and Lynn, over Google Hangout. We discovered the undeniable passion and determination that drives our companions at the Bura Girls School. We were impressed by their knowledge and commitment to the project and to their studies. We had the opportunity to ask them questions about the topic of malaria and about themselves. Their perspectives were refreshing and gave us much needed knowledge to understanding malaria firsthand. As a world leader, the United States has the responsibility to help other countries fight the disease. Preventative measures can lead to a healthier society and consequently a more productive economy. There is no one treatment or prevention that will eradicate malaria. It's going to take many approaches, countless researchers, and a multitude of inspired minds. Education of malaria is important within the affected communities as well as those who are not affected. People who are interested in helping can take many approaches. Helping those with malaria can be as simple as monetary donations to preventative organizations to as large as becoming a scientist or researcher in the field. This project has inspired us to help. We decided to start a fundraiser for science and laboratory supplies for the Bura Girls High School.